Okay, good afternoon, everybody. I am supposed to talk about neuroplasticity. And first of all, I want to introduce myself. I'm head of the bioimaging lab. Here is the team at a certain stage in time because people are coming and going. And we are focusing on experimental MRI, and this will also be reflected in my talk. So we have 3 small animal MRI systems, and in Antwerp there is a building entirely devoted to small animal imaging. Part of it belongs to the bioimaging lab, where we can do MRI of small animals. These are the animals which are our target, so we go beyond rats and mice. We go to birds and fish, and I will mention some of the bird work because it's a very nice model for plasticity. And at the other side of the building, there is another team which gives access to small animal PET and SPECT. So actually we can do anything on small animal imaging. So I was asked to talk about neuroplasticity, and this is the outline of my talk. First of all, I want to mention the definition, that it involves critical periods, that it is important in both health and disease. Then I will talk a little bit about the key mechanisms, and then I will show how you can use imaging to monitor neuroplasticity. The definition of neuroplasticity, I think each of you has a sort of idea what neuroplasticity is about. It's actually the brain's ability to reorganize its connections functionally and structurally in response to changes in environmental experience. I think learning is the best example. Most of you think when they hear neuroplasticity in terms of rejuvenation or recuperation from stroke or any other insult. This is also neuroplasticity. Neuroplasticity is dealing with critical periods. It means that in life there are certain time windows, critical periods, during which neural circuits display a heightened plasticity. And later on, the plasticity dramatically wanes. For instance, early postnatal life is a critical period during which neuroplasticity is very high. I have a pointer here, I see. So, postnatal life is associated with high plasticity. After that post-critical period, there is still plasticity, and in adult stage, the plasticity has dramatically decreased and can be considered low. When I mention neuroplasticity involves health and disease, you could say that plasticity is a natural process, but that plasticity can be modulated to recover from a neuropathology, and the neuroplasticity can also be modulated and cause and induce pathology. Now, the key mechanisms which are involved, and I will briefly explain them to you, are Hebbian plasticity. So we're dealing with excitatory neurons, for instance, pyramidal neurons. Second part will be intracortical Gabaergic inhibition, dealing with Gabaergic interneurons. And finally, I will talk about new neurons, neurogenesis. Those are 3 major aspects which deal with neuroplasticity. And here is a picture of Donald Hebb, who found out about Hebbian plasticity. We call it a Hebb's postulate. And you know that in the brain different neurons talk to each other through action potentials. And Hebb's postulated that when an axon of a cell A is near enough to excite another cell, and he does this repeatedly or persistently, then some growth processes or metabolic changes take place in both cells. This deals with plasticity. So when you have a repeated or persistent firing of neurons, and we call this long-term potentiation, then here's a pointer, okay, which is shown here, So actually what is shown here are local field potentials, but you could look here that when you do repeated stimulation from one neuron firing to another one, for instance, or you do this electrically, of course, then you can see that you have an increase in activity in the next neuron. When you do this persistently, you induce growth processes or metabolic changes. And this is what's shown on this slide. So, I have to go back. So what you see here is a synapse. And after long-term potentiation, so repeated stimulation, you see changes occurring. For long-term potentiation, you need presynaptically glutamate to be released in order to have a polarization postsynaptically, and you need more receptors. And I mention here glutamate receptors, AMPA receptors, and NMDA receptors. And what is changing is the amount of glutamate can change, or the amount of, this is dying, or the amount of receptors, NMDA receptors or AMPA receptors, you can see on the picture. Okay. So these are metabolic changes which occur after long-term potentiation. And typically, what you can see is that, what is known is that there is spine formation, resulting in new excitatory synapses. So what you see here is that this is a synapse and a postsynapse, and you see changes, metabolic changes, which deal with increasing amount of receptors, or like glutamate receptors, other compounds. I'm not going into detail. And the surface of the synapse the synapse enlarges, and in the end, next step is that there are new spines formed, spine pruning, and this is what you can see here on the next one. I can't show it, but you can see it on the picture next to it. Result of, okay, thank you. Result of this spine pruning is shown here. So Hebb also postulated that the more often neuronal pathways fire, the stronger the connection will become. This long-term potentiation is the basis of learning and memory formation. And this resulted in his famous quote, which is mentioned here: neurons that fire together, wire together. And that's the principle of neuroplasticity. And the best model, the best studied model also, is hippocampal learning. Now here is an overview of molecules that are involved in the changes that occur at the level of the synapse or the postsynaptic ones, which deal with different types of molecules. And I'm not going to give you all the details, but I think it's obvious that there are several diseases like schizophrenia in which there is impaired Hebbian plasticity because one of the molecules is either down or upregulated. So a lot of transgenic mice can be made to investigate what's going on during long-term potentiation and Hebbian plasticity. For instance, if you provide NMDA antagonists, you would prevent Hebbian plasticity. If you have knockouts of some NMDA antagonist or molecules, you could find out what role it plays in these plasticity events. Okay, so far Hebbian plasticity dealing with activity-induced changes of excitatory neurons. Next part is intracortical Gabaergic inhibition. Here I'm talking about interneurons, a specific part of interneurons. And it's obvious that inhibitory Gabaergic circuits can play an important role to steer and modulate the activity that's going on in the brain in certain circuits. And most people who have studied this intracortical Gabaergic inhibition have focused on the visual system in young cats or in mice. The visual system has been studied a lot for neuroplasticity. And this is an example of a human being, of course, but in case you're studying a mouse or a young cat, during the critical period, so postnatally, typically what is done, one of the eyes is covered or occluded. And what will happen? The projections that result from the eye that is covered will decrease, and the eye that is left open, its projections will increase. This is typical neuroplasticity, but only occurs in a short time window postnatally. If you wait longer and you cover the eye, the effect will not be as high as during this period. And people have studied this to discover what was important. Here I give you a schedule of intracortical Gabaergic inhibition, and I will explain a bit. So this is the neuron I was talking about when I mentioned Hebbian plasticity. This is an excitatory neuron, and here are different types of interneurons that they don't have spine formation, but they have boutons, it's a different name, and they will influence this neuron by Gabaergic inhibition to sort of modulate the amount of activity versus inhibition. It's a sort of balance. And this is a very critical balance. And in order to have neuroplasticity, you have to have a certain degree of Gabaergic inhibition neurons like this one. This is shown, for instance, by using transgenic mice, which lack some very important components in Gabaergic expression, no GABA, for instance. Those animals will never develop an ocular dominance if you cover one of the eyes. Neuroplasticity is not operational. So this is very important, and that's how they found out that Gabaergic inhibition, the role of the interneurons in inhibiting the excitatory neurons to some extent, is very important for neuroplasticity. This schedule actually shows the degree of cortical plasticity depending on the amount of Gabaergic inhibition. So this is postnatal age, and you can see on average that there is a maximum in plasticity, and at that moment your Gabaergic inhibition is sort of medium. You need a certain amount to have neuroplasticity, and when the Gabaergic inhibition becomes too high, the plasticity is finished. So you can modulate this Gabaergic inhibition to open your window of postnatal neuroplasticity, that it starts earlier. This is what happens in transgenic mice who have BDNF overexpression, for instance, or some therapeutics. Or you can postpone your neuroplasticity. So this is, you modulate the Gabaergic in strength, and this allows you to change the window of neuroplasticity. So this is actually the same, but here you can see the Gabaergic inhibition expressed here, and in green is what happens when during youth, so postnatal plasticity. When you have normal adults, the inhibition will increase, and with environmental enrichment or with some antidepressants, you can increase the plasticity again, so you can come back to the situation of juveniles. And then, of course, when the cortical inhibition is results in an over-inhibition, you have some type of psychiatric disorders which deal with mental problems. An example is mentioned here: Down syndrome, Rett syndrome, but also autism spectrum, schizophrenia deal with over-inhibition as a sort of disease pattern. This picture also shows that there could be some therapeutic efficacy by environmental enrichment. I will not go into this, but this is actually showing the same, and also that with some treatments, with some treatments you can modify this plasticity. Third important part, new neurons. Early in postnatal life, we know that from our children, they first see, then they start to walk. This all deals with how your brain further develops. And what is shown here is regions where new neurons sort of emerge, they migrate, and they develop into new neurons. Now, in adulthood, there are two parts in the brain, two places in the brain at least, where neurogenesis is still going on. And these regions are the dendrite gyrus in the hippocampus and the subventricular zone lining the lateral ventricles. And this one is related to the rostral migration stream, which is providing new neurons in the olfactory bulb. Both stem cell niches are involved with making new neurons in part of the brain which need them for making new connections, dealing with memory or with different smells. Okay. This is another view where you can see the stem cells positioned in the subventricular zone. Okay, so neurogenesis is a natural event which decreases in age, but is still present in adult animals and humans. But of course it's influenced by a variety of growth factors, cellular matrix proteins, pathologies. For instance, inflammation is affecting it. And it's also demonstrated that in neurodegenerative diseases, the topic of the previous speaker, that typically the subventricular zone is affected. The neurogenesis in the subventricular zone is affected. Neurogenesis is also affected in depression at the level of the hippocampus. And antidepressants, some of them upregulate neurogenesis in the dendrite gyrus. So neurogenesis is something which is of quite importance. Okay, after this short overview of neuroplasticity and what it means, I would like to show you how you can image neuroplasticity. And the focus will be on MRI, because I think MRI is providing most of the tools to do this. So as I said, neuroplasticity is a natural phenomenon that is impaired in several diseases, and so it's gaining interest that we try to image it and quantify it, either as a diagnostic or a treatment follow-up tool, or to understand the underlying mechanisms and then use them later on for treatment. The first example I will show you is how functional MRI can be used to visualize long-term potentiation-induced neural network reorganization. I will show the work done in Logothetis lab in Tübingen, where they actually measure synaptic plasticity with fMRI. First of all, I will explain briefly what functional MRI is. If you have some neurons which are activated, they require more oxygen and more glucose, and as a result you have a vascular response, meaning that there is more blood coming to the region which is activated. There is an increase in CBF, cerebral blood flow, and cerebral blood volume, and as a result you will see there is a change in the amount of oxygenated blood, and the amount of deoxygenated blood will also change. And this is what we call the bold response. It's the blood oxygenation level dependent contrast, which is based on the fact that there is a vascular response to activated neurons, and this is how it looks. This is the activation, and this is the overshoot that you get. Now this is an example where this is a rat brain, and you slice it from the bottom to the top horizontally. These are control rats, and you stimulate them. The arrow is indicated here. You stimulate them here electrically in the magnet. This is done. This is very nice work. And this is an animal which has been stimulated, which has been potentiated. So it means that you have high frequency stimulation. And then you stimulate in the magnet at the same time. The difference is that here you have hippocampal stimulation, and you see that it crosses the hemispheres. If you do this in an animal which has been potentiated, meaning submitted for a few hours to repeated stimulation, you will see that the circuit not only involves the hippocampus, but also expands, like the entorhinal cortex, prefrontal cortex, and all these areas which are shown here. When they do the same experiment with NMDA antagonists, it didn't work. So this is showing how long-term potentiation increases your circuitry, so involves more neurons in its activity, and that you can show this with functional MRI based on hemodynamic responses. Example 2, I was talking about neuroplasticity of the visual system, ocular dominance, and I said that in small cats or in mice, you can cover one eye and then see that the projections from that eye will diminish and the projections from the eye that is open will increase. I show you some data from a natural model. This is a bird in an egg, and during development the bird is folded in a standard way so that one of the eyes, the one which is looking at the shell, during a critical period receives light, while the eye which is lying on top of the body is actually sort of deprived from light. And actually birds, when they hatch out of the egg, they already have ocular dominance. And this is because of that the fact that one eye was exposed and the other one wasn't. So if you do fMRI, this is an example of fMRI where, this is an example of fMRI where, and let's focus on this layer. So these are bird brains, of course, and you can do binocular stimulation. You can only stimulate the left eye or only the right eye, and you can see already when you make the difference. If the right eye has a projection, of course, on the left side, you can see that there is a left dominance, a right eye dominance, so a left side in the brain which is more activated. This is how you can change, how you can study ocular dominance, and also could study how you could manipulate it pharmacologically to see changes. Okay, these were two examples of how you could use functional MRI to study long-term potentiation and plastic changes postnatally. The next example is dealing with another MRI method, which is diffusion tensor imaging. And we used it in the lab to assess seasonal rewiring of the brain in seasonal songbirds. So I have to explain a bit the songbird as an extreme model for neuroplasticity. And as I mentioned to you that plasticity deals with critical windows, in seasonal songbirds, the neuroplasticity critical period reopens every spring. That's what is making it a nice model. Okay, so this is a short explanation about the songbird brain. Songbirds have a system in the brain, a song control system, which is important for singing, of course. The system is highly plastic. Most of you know that the males sing, the females don't. Females treated with testosterone sing, so it's highly testosterone driven. And birds sing in spring, they don't sing in summer. And in spring, the song control nuclei responsible for the singing change in volume and in connections. And singing is a learned behavior, so learning and Hebbian plasticity, it all comes back. Here is an example of two types of birds. This is what I call a limited learner, this is an open-ended learner, and let's focus on this one. We can see that each spring he can learn again and upgrade his repertoire. So it's called the sensory phase. The critical period for learning reopens each spring, and in the brain you will see dramatic neuroplastic changes as well. Of course, we've worked a lot on this in my lab, and this is a paper which shows you that there's a lot to tell about what you can do with MRI when you want to study neuroplasticity. This is an example of how the bird brain looks, and of course it's much smaller than the human brain. Although it looks different, there are a lot of similarities. A little focus. This is a schedule of a bird brain, and you should only look at this schedule, which is the song control system. And in summer it looks like this, and in spring you will see that the volumes are larger and the connections are tighter. So neuroplasticity deals with making new connections, with incorporating new neurons, with selecting a balance with apoptosis, so all the aspects play a role. Okay, so I will show you how we use diffusion tensor for this. When you look in the brain, you can have typical, like this, you have a cortex structure, but sometimes you have white matter structures, and in that case, diffusion tensors plays an important role. What I show you here is how water in cells and extracellularly is displaying a free motion, and the motion is happening at random, so it goes in all directions the same way. This is what I would call isotropic diffusion of water. In some cases, when you have fibers, then the motion of water will be restricted rather in one of the directions, and then you have anisotropic diffusion. And based on this, you can have a contrast in your image, and you can visualize fibers. And this is an example that you can imagine that the water diffusion will be larger in this direction and the opposite direction, and you can draw conclusions on increase of axons or demyelinization or things like that, provided you do the proper histology. Now, this is how it looks when you look at a bird brain. They don't have a lot of white matter, so it doesn't appear at the T2 weighted images. Here is an FA map where the colors display the direction of the fibers. This is cerebellum, and this is the optical fiber, the optic chiasm actually. So you can display these fibers in all sorts of manners, like with small cigars or with fiber tracking, whatever. This is what you often see happening in human DTI images as well. Okay, so what we typically do is we measure birds repeatedly in spring and in summer. Then you average a typical summer brain and a typical spring brain, and then what you could see here immediately is that there is a change in volume of the entire brain. We only looked at one half, but it doesn't matter, and individually it also changes. And you can look at statistical changes and find out where there are different DTI parameters. And I mentioned just a few. I'm not going into details. I have to keep an eye on the time. I can continue for 5 minutes? Yeah, because I started later. Okay. Okay, so what you can see here is how the optic chiasm, for instance, is changing seasonally, as a nice example. And here you can see how the connections of the sound control system also become tighter in spring as compared to summer. So you can really visualize seasonal rewiring of the brain, and you can quantify it. This is another example of fibers that go lower in the brain and are responsible for singing. Okay, so structural rewiring is something we could do, but initially we worked a lot with manganese-enhanced MRI. And manganese-enhanced MRI is also used for studying neuroplastic changes. And I will explain a bit. Manganese is a biological calcium analog, and when you inject it in the brain, for instance, you make sure that this neuron has access to manganese, it will pick up manganese instead of calcium and transport it through the axon, release it at the synapse, and it can be transported transsynaptically. It's used as a measure for axonal transport, actually. So if you inject in the brain, the neurons that are here and project to that region will be visualized. And this is what you see when you inject here. You can visualize this region and also the other one, so you can follow projections. And you can follow this in the magnet by doing the injection with a long tube from outside the magnet. And then you can sort of do what I call dynamic manganese-enhanced MRI. You follow the manganese uptake in the nuclei, depending on which one you want to study. And depending on the activity, you will see changes. And this is an example where you treat females with testosterone, so you masculinize them. Let's look at this situation. The nucleus not only increases in volume by 50%, but also the uptake slope is changing, reflecting a change in the functional connectivity. And just to remember, these are the same circuits which we could study with DTI to see changes in the anatomical connection. The manganese is reflecting some functional connection which you can quantify and which is related to activity. Of course, same type of tools are used, for instance, to study massive fiber plasticity in epileptic models and study abnormal fiber wiring in all sorts of other diseases. Okay, then another tool you could use is resting state fMRI. And I will explain a bit. I already told you that when you give a certain stimulus, with functional MRI you could observe a bold response. Now, even without the stimulus, there are sort of resting state fluctuations of that bold signal. And for instance, when you look at this work of Biswal, this is on the one hand functional MRI when you give a motor stimulus, and this is resting state, only showing fluctuations of the same circuit. So it's actually showing you which regions in the brain show the same low frequency fluctuations and are thus functionally connected. Now, we've set up the method in rats and in mice. And so what you see here are components, and these components are regions which display the same low frequency fluctuation, which is translated in electrophysiology as local field potentials. Okay, I will not go into that, but if you look at human literature, resting state is used to study motor learning, motor skills, learning languages, to study interhemispheric plasticity, because you can visualize and quantify functional connections and how they change. And we try to do this in birds, and I will give only one example. So you acquire bold images, which takes only 5 minutes, and the focus is still on the same circuitry which I showed before. And then you can see, for instance, that in fall I can see part of the circuit, and in spring it is connected with other parts of the sun control system, just as an example. Okay. So conclusion, functional connections during rewiring can be followed with resting state function MRI. And then finally, neurogenesis is also something you could study using MRI. And I show you here briefly how we do it. This is the schedule I showed before. What you typically do is inject in the ventricles contrast particles, which have iron to visualize on the MRI, and which have fluor to do some histological validation. And then you can monitor the cells in the subventricular zone pick up the contrast, and as they migrate along the RMS, you can follow them up to the olfactory bulb. So this allows you to monitor neurogenesis or impairment or improvement of neurogenesis. The same thing can be done with bioluminescence imaging. Then you don't inject a particle, but you transvector the subventricular zone with a vector expressing luciferase, and you can follow migration of young neurons to the olfactory bulb with bioluminescence imaging. Okay. That's it. I show how you can validate this with histology, but this is not important. It's important, but I have to finish. So in vivo monitoring of neural progenitor cells could be important for studying neurodegenerative diseases as well, or the effect of antidepressants. Also, GABA has an influence on neurogenesis, and I mentioned GABA in another context before. And MRI has a high spatial resolution to do it, but I showed you the BLI images because BLI is a viability marker, and it allows you to tell whether the cells are still alive. Okay, I finish here. Thank you for your attention.